The last group of macromolecules that we're going to talk about are the nucleic acids. And if it seems like there's less information for this group than the other ones, have no fear. The reason why we're giving a little less information right now on this group is because we're going to talk about nucleic acids later in the semester more than any of the other groups. So they're getting a little less lip service now, but in the scheme of things, don't worry, we'll get plenty of information on them. So nucleic acids, like proteins and like carbohydrates, the molecules that are classified as nucleic acids are all grouped together because they share a similar base structure. They, like the carbohydrates and proteins, have a very repetitious structure comprised of monomers that make up polymers. The building blocks or monomers of nucleic acids are called nucleotides. And like the other macromolecules that show this repetitious structure, the monomers themselves have a very specific structure. Nucleotides always have these three components. They have a phosphate group, which, as we've talked about before, is a functional group. Pictured here, a phosphorus that's bonded to 4 oxygens. They have a sugar, the type of which varies, but irregardless, they have a ring-like sugar that is shown in this image right here. A monosaccharide, to be specific, so a single sugar. And then they have a nitrogenous base. There are 5 different nitrogenous bases that exist. However, the different types of nucleic acids might not have all 5 of those present in them. And we'll talk about that in a moment as well. So every nucleotide has these 3 components, and essentially the polymers of nucleic acids are made of these nucleotides being bonded over and over and over together. So for instance, if we look up here, here's one single nucleotide. And if we look at the larger structure as a whole, we can see that the phosphate group and the sugar sort of make up the backbone of the polymer. And then the nitrogenous bases extend from that backbone. And the entire polymer is just a compilation of many of these nucleotides bonded to one another. There are 2 types of polymers, so 2 major types of nucleic acids. That is DNA and RNA. Now, they obviously are similar in that they are comprised of nucleotides, but there are sort of 3 major structural differences, and then we'll talk in a moment that there's also functional differences between these 2 types of nucleotides. Let's go through the structural differences first. The first structural difference is actually illustrated in the name. So DNA is actually just shorthand for deoxyribonucleic acid, whereas RNA is shorthand for ribonucleic acid. That deoxyribose versus ribose, those are different types of sugars in the nucleotide. So we said that nucleotides have a monosaccharide sugar that's in the form of a ring. And that's true of all nucleotides. But the 2 different nucleic acids have different types of sugars in the nucleotide. Specifically, DNA has deoxyribose sugar, and RNA has ribose sugar. I just want to point out, remember, the suffix -ose is an indication of a carbohydrate. So these are those single sugars that differ. That's one structural difference between DNA and RNA. We then have a difference in number of strands. So how many strands? RNA is a single-stranded molecule, and DNA is double-strand. So RNA is just a single phosphate group sugar backbone with one nitrogenous base extending from each one of those nucleotides. On the flip side, DNA is double-stranded, which means that it has 2 phosphate sugar backbones that run parallel to each other, and then 2 nitrogenous bases extending towards each other that are connected via hydrogen bonds. So let me kind of draw on what this might look like on this picture. Give me a minute. My art skills leave something to be desired. So we would have on the opposite side a sugar phosphate group. Extending from it would be a nitrogenous base. And the nitrogenous bases are connected via hydrogen bonds. And then likewise, we would have a sugar, phosphate group, attached to another nitrogenous base that is connected via hydrogen bond to the nitrogenous base of the other side. Now with DNA, these strands end up getting twisted into the form of a helix. So a lot of times it's described as like a twisted ladder. Within the ladder, the sides of the ladder are the phosphate groups and the sugars, and the rungs of the ladder are the nitrogenous bases that are connected from either side by a hydrogen bond. Whereas RNA is kind of like half the ladder. It doesn't have the other side. The third difference is the types of nitrogenous bases present in the nucleotide. They both have adenine, guanine, and cytosine. So those three nitrogenous bases are universal between DNA and RNA. But what differs is what their fourth nitrogenous base is. DNA has thymine as its fourth nitrogenous base, and RNA has uracil as its fourth nitrogenous base. There is no uracil in DNA, and there is no thymine in RNA. Those two get swapped out in their respective nucleic acids. So those are the three structural differences between the two types of nucleic acids, DNA and RNA. Now let's talk about functional differences. Of these two, people are usually most familiar with DNA, so we'll start there first. DNA is essentially the master copy of all of the genetic information. And when we say information, specifically this information is instructions for all proteins. Now, it's essentially instructions for how to build every protein that living organism is capable of producing. Okay, I always call it sort of the master copy. It's got information for everything. And this is what gets passed down from parent to offspring. So if you have children, then you pass down 50% of your DNA to your kids. Hence, what causes resemblance, which we'll talk about in a later chapter. Now, RNA. RNA is a copy of a single protein. So it's a copy of instructions made from DNA for a single protein. We're going to have an entire chapter dedicated to talking about how proteins get manufactured, okay? But during the manufacturing process, you need to have instructions for building the protein. The master copy of instructions isn't what actually gets sent to the protein building factory. The DNA gets held within, at least most organisms, the nucleus so that it stays nice and safe, which again, we'll talk about in later chapters. We're just kind of setting the stage here. So the master copy of the DNA has to remain intact, and we don't want it to get damaged. It's not what gets sent to the place where proteins are manufactured. Instead, the cell makes a copy of instructions of just the protein it's going to build. That copy is RNA. So RNA is a copy of instructions for a single protein that gets made from DNA, which has the instructions for all of the proteins. And that's what the protein actually gets built off of, is the RNA, which is a copy of the protein from DNA.